Hello. Hello, I'm Elena King, and today I will be giving a presentation about biologic oxidation, chapter 12. So before we go deep in, I would like to start with some definitions. So first of all, oxidation means removal of electrons, which is basically losing electrons. And for reduction, it's the opposite. It's gaining the electrons. So these two reactions are always coupled, which means they always occur together, and this is the basic mechanism in ETC. So as you see in this diagram, you can see A is being oxidized and B is being reducted, and these two reactions are and coupled together. In biologic oxidation and respiration, like many oxidations can occur without oxygen, and this is actually the example of dehydrogenation. But in respiration, the oxygen is very crucial here to produce ATP because it is the final electron acceptor and it is essential for ATP synthesis, which is related to energy production and cell survival. As in this redox reaction, hydrogen and water or they react together and finally form water as a final product. Oxygenases is the enzyme that uses oxygen as a substrate, and cytochrome P450 is the example of this. So this cytochrome P450 metabolizes drugs and toxins, and this is important because this oxygen therapy is critical for patients who usually have respiratory failure or circulatory system problems. Next, I will move on to free energy and redox. So this free energy is basically a tendency of molecules that to donate or accept the electrons. So this tendency of accepting and donating electrons are usually written as E0, which measures this tendency. And the standard biologic conditions here is pH 7, and this is equivalent to hydrogen electrode at 0 - 0.42 volts. And this kind of information helps us to predict the flow of electron and metabolism rate. Oxidases is an enzyme that transfers electrons to oxygen and makes waters or hydrogen peroxide as final product. And cytochrome oxidase was introduced in previous slides, which is equivalent to ETC terminal enzyme. And these are the final electron acceptor that accepts the electron and produce water or hydrogen peroxide. And this is inhibited by some toxic substances like monooxidase, H2S, and cyanide. Flavoproteins has this, they contain like special cofactors of FMN or FAD, which originated from riboflavin. And they also participate in redox reactions, and during that reactions, they often form intermediates as semiquinones. Dehydrogenases is one of the most common theme here. So in general, it transfers the hydrogen between molecules. And the most used coenzyme here is NHD plus or NHDP plus. And these are involved in oxidation and reduction because these substances actually work as electron carriers that traps the electron and release when it should be released. So as I talked before, NHD and NADP are the most used coenzymes here. So if we focus on NAD plus is reduced as NADH, and this NADH has the electron that can be released as the electron receptors, which is basically related to energy productions. And this system are very oftenly found in glycolysis and TCA cycle. NADP plus is also used in biosynthesis as well, usually in fatty acid synthesis and pentose phosphate pathways. This diagram shows that NHD redox reactions, like how NHD plus accepts hydrogen and NHD plus accepts electrons and become NADH. And later on, it will release the electrons at its electron acceptors. So ABD and cytochrome. Some enzymes use ABD or MN, and these are tightly bound to proteins that so it can function as electron carriers. And cytochrome here also does the same thing. It works as electron carrier, but it does that by containing iron here. So this diagram shows like how NHDH becomes NHD plus by releasing electrons, and this complex is 1, 2, 3, 4, and redox is happening here, which ends up in proton gradient, which is the crucial point in ATP synthesis. Hydroperoxidase and ROS. So even though like oxygen is a crucial point here in cell respiration because it acts as a final electron acceptor, but this oxygen has a very toxic species that can cause cell damage if they're accumulated. So hydroperoxidases are the enzyme that protects cell from this toxic oxygen species. And so it basically protects cell from ROS damage because ROS damage can cause DNA, protein, and lipid damage. And the examples for this peroxidases are peroxidases and catalysis. So peroxidases use glutamine or vitamin C to detoxify hydrogen peroxide. And catalyst converts this peroxide into water and oxygen, what is less toxic. And this catalyst is very important because it is a very fast enzyme, and when cells are doing respirations for its survival, the efficiency is very important here. So catalyst is one of the most important enzymes in biosystem. Oxygenases are also the enzyme that has oxygen as a substrate. So there are two types of oxygenases versus dioxygenases, which is adding both enzyme, and another one is monooxygenases. It is adding only one enzyme, and another one is turned into water. Cytochrome P450 is a major example, of monooxygenases system. This is located in liver, and it has its main role in detoxifying drugs and chemicals, but it is also important because it also works as making those substances and more water soluble, so it will be easier for them to excrete it. And this is also involved in steroid and vitamin D metabolism. Superoxide is also a very toxic species of oxygen, but this one is reactive as a free radical. And because it is very reactive, it causes cellular damage. And SOD is an enzyme that converts this toxic superoxide into peroxide and oxygen too. And peroxide here is also toxic, but less toxic than superoxide. So this peroxidase will be detoxified later in catalysis or peroxidases later on. So this diagram shows how the superoxidase is converted to peroxidase which will be detoxified later on by SOD. And this is also used in cell signaling. So as I said, even though peroxidase is an already detoxified form of superoxide, it is so toxic, so it should be removed by catalysts or peroxidases. And this detoxifying processes are actually preventing this RS accumulation. So it basically protects cell from damage because the accumulation of toxic oxygen species can cause cell to die. So in conclusion, this reaction, redox reaction produces ATP. So the oxygen is crucial here because it actually contributes a lot in this energy generating processes, but it also generates these harmful auras that can actually damage cells. So this should be handled by enzymes, so it can be detoxified. So the cell survival is basically the balance between these these reactions of ATP generating and detoxifying. So if we move on to the five multiple choice questions, the first one, which of the following best defines the oxidations in biologic system? So this was the basic definition question. The answer here is loss of electrons. And this should be coupled with reductions gaining the electron to match the overall balance in cell living system. Which enzyme is a terminal component of electron transport chain that transfer electron to oxygen? The answer is cytochrome oxidases. So cytochrome oxidases was introduced often throughout the presentation. So it acts as a final electron acceptor, and it is important here to generate ATP and make water as a final product. And it ends the overall ATP generating processes. It finishes it. Yeah. Which coenzyme is primarily involved in oxidative pathways, such as glycolysis and citric acid cycle? The answer is NHD plus, because NHD plus accepts electrons, become NADH, and it releases this electron to electron acceptors, which will also contribute into proton gradient and the overall cell respiration process. So the NHD plus will be the answer for that one. What is the primary function of a catalyst? So the answer here will be C, break down hydrogen peroxide into water and oxygen. So what is the main role of the peroxidases? Since hydrogen peroxide is a very toxic form of oxygen, it is broken by enzyme into water and oxygen, which is not toxic. Which enzyme protects cells by converting superoxide into hydrogen peroxide? The answer here is superoxide demutases, which is introduced as SOD in the slides. So it converts superoxides into peroxides, and this peroxides will be detoxified into water and oxygen again. So there's a basic detoxifying process in the cells. So these are the sources that I use to prepare for these presentations. And that will be the end of my presentations. Thank you so much.